unwanted environmental microbes. And this truly is one of the most important techniques you guys can take out of the microbiology lab. If all you learn is really good aseptic technique, that's huge. You're going to save patients' lives having really good aseptic technique. Okay? So the way we wanted you to check to see if your aseptic technique was good is we wanted you to check your slants for evidence of contamination. Now, what you'll be looking for on your slants is, first of all, you're going to look at the surface of the slants where the microbes were growing aerobically. And we chose, let me just back up here, I'll make sure I get the microbes right. You will recall that you transferred Micrococcus luteus to a regular TSA slant, and you streaked the surface of the slant. Now, the reason we chose Micrococcus luteus is it makes a beautiful yellow pigment. So one way to check to see if you had good aseptic technique is to check the color of the colonies on the surface of the agar. And hopefully they all appear yellow. You don't see different colored colonies growing in there. If you do, it suggests you had a break in your aseptic technique. Maybe you forgot to flame your loop. Maybe you left the cap off too long. Maybe after flaming the loop, you accidentally touched it, maybe to your lab coat or the bench. So we're looking for colonial growth that looks all the same, not multiple colors in there. Now, on the big butt slant, you guys will recall we used the stab and streak technique. We transferred E. coli that was growing on a slant. Using our inoculating needles, we stabbed the butt to get the E. coli growing anaerobically to force him into fermentation. And so that was our stab. And then you streak the surface so that the E. coli could grow aerobically. Now, E. coli is a non-pigmented organism, so you'd expect to see the colonies appearing white, kind of a whitish color, non-pigmented. But again, you want to check if you have multiple colors of colonies growing on the surface, somewhere you had a break in your aseptic technique. The other thing we wanted you guys to check for is that in the big butt agar deeps, in the butts themselves, hold them up to the light, and you're looking for evidence of cracks or bubbles. And if you see cracks or bubbles, that's telling you your bacterium carried out fermentation with production of gas. And the gas is usually either molecular hydrogen and or carbon dioxide. And next week when we start doing metabolic tests, the ability of your microbes to produce gas during fermentation will become important in helping to identify them. Okay, so did anybody get cracks or bubbles in their big butt agars? Yeah, and sometimes it's kind of subtle. Some people think, oh, I didn't think that was significant, but a little crack, a little bubble, that's significant. Yeah, so your E. coli should have produced gas. Your E. coli should have produced both molecular hydrogen and carbon dioxide. So we would predict we'd see bubbles or cracks in those big You see it? Good. Good. E. coli read the lab manual. That's what we want, is microbes that read their lab manuals while they're growing in the incubators. Good, you guys. Okay, so the final one we're going to take a look at is the broth tubes. And you guys will recall that you transferred Serratia marcescens into your trypticase soy broth. Now, we incubated at 30 degrees, and Serratia marcescens at 30 degrees makes a beautiful red pigment. And you can see the color of your broth has that beautiful brick red pigment of Serratia marcescens. Now, we don't want you to disturb the tubes because we want you to make note of where the cells are growing. And we want to introduce you to some conventional terms that are used to describe the patterns of cell growth in broth. So if you have a collection of cells that are growing at the air-liquid interface, that's called a pellicle. And it can be described as a type of biofilm. It's not attached to a surface, but it's attached to this air-liquid interface. And some biologists say it may suggest that your organisms do carry out aerobic respiration. We want to remember that oxygen diffuses poorly through water-rich environments, so that, of course, the maximum oxygen concentration is going to be up at the air-liquid interface. Now, next what you want to do is hold your tubes up to the light and search for a collection of cells at the bottom of the tube. That's referred to as a button. That's referred to as a button. Now, some microbes, when they grow, they all settle to the bottom of the tube. So when you pull your tubes out of the incubator, you might think, oh no, I did something wrong. My microbes didn't grow. That's why you always want to hold the tube up to the light, and it might be you did a great job, just all your bacteria settled to the bottom of the tube. Okay, so that's called a button. And then if you see cloudiness, that cloudiness is referred to as turbidity. And it tells us cells are present. We don't know if they're alive, we don't know if they're dead. All we know is cells are present. Now here's a real important thing, you guys. Just looking at your tubes right now, do you have any idea whether they're pure or mixed cultures?
No.
Yeah.
No. Yeah, there's no way in the world to know, right? So that's a disadvantage of broths. Just looking at them, all you know is cells are either present or they aren't present. But you have no way of knowing if the culture is pure or not. Okay, so that's an important thing to remember. Okay, so I'm just going to keep pushing us on, you guys, so we'll finish the discussion and then you guys can start recording your observations. So the question is, if I had a broth culture like this, how could I determine if it was contaminated or not? Well, one way would be to take a loop full of your culture and streak it onto an agar plate and get isolated colonies. If you get isolated colonies and you see two very different colony types or 3 or 4 different colony types, that suggests you have a contaminated culture. And the reason is, our presumption is different microbes will form different types of colonies. So what we're going to do last is we're going to have you guys take a look at your agar plates. And you incubated one plate at 30 degrees Celsius, okay, and one of the organisms I believe I gave you in the mix was Serratia marcescens. So at 30 degrees, the Serratia marcescens should be making beautiful reddish, brick-colored colonies. And then the other plate we had you incubate at 37 degrees Celsius. Now this is so cool. Pigment production in many microbes is temperature dependent. Serratia usually turns off pigment production at high temps, like 37. Okay, so the Serratia growing on your 37 degrees Celsius plates, they might not have much pigment, or maybe they're only like a faint pink color. And I think the reason was we turned the incubator off yesterday, so they had a little bit of time to grow it at lower temperature. The cultures you used, they were mixed cultures. So we had Serratia marcescens, and then we had a micrococcus as the second organism there. And so what you want to do is you want to check for evidence that you had a mixed culture. So you guys remember we had the 4-quadrant technique. One thing to remember in mixed cultures, often one microbe is present in far higher numbers than the other one. Okay, so let's say, let's pretend this represents the Serratia. And the Serratia, let's say, was present at really, really high numbers. So maybe it wasn't until quadrant 4 that you got isolated colonies. Okay. Now, a mistake some folks make is when they look at a plate, they'll only look at the last quadrant in which the colonies are growing. And if they look at the last quadrant and say, oh, I only see one kind of colony, my culture is pure, that's a big mistake. Because often contaminants, as we said, are in lower numbers. So you might not see the contaminants in the last quadrant, you might see them in a previous quadrant. So whenever you're inspecting your plates for contaminants, it's very important you look at all 4 quadrants. Often a contaminant is hiding in a previous quadrant. And you want to look both at the surface of the plate and the bottom of the plate. Because very often, one microbe will overgrow a second microbe. But you can often see the second microbe if you flip your plate over and look at the bottom of the plate. Okay, now what's the big deal about isolated colonies? Well, our presumption is that these isolated colonies that are not touching one another, the presumption is they all arose from a single microbe. A single microbe was spread onto that portion of the agar. They absorbed the nutrients, they grew, they divided. Now a single colony can have between 10 to the 8 to 10 to the 9th cells. Okay, and we presume they're all descendants of that first cell. So each isolated colony represents a pure culture of microbes. Pure culture of microbes, single genetic type of microbe, right? So the question is, if you have a contaminated culture or a mixed culture, how can you, if you have access to a lot of fresh agar plates, how can you get your 2 different microbes? Let's say you've got this Serratia microbe here, and let's say the little white guys are your micrococcus. how could you get them into pure culture if you have access to more plates? What could you do? Something pretty simple. So right now we've got a mixed culture right here, right? And we want to get the 2 different microbes into pure culture. What we do is we just get two new plates, right? We get 2 new plates. We use aseptic technique to transfer cells from, say, an isolated serratia culture. We'll transfer those cells to a new streak plate, streak them for isolated colonies. Okay, so we'd transfer cells from there. And then we could go and hopefully find an isolated colony of the second organism, say our micrococcus, and we could transfer the micrococcus to a second plate. And hopefully within 24 hours after incubating these plates, we will have separated the 2 organisms so that now we've got a pure serratia culture and we have a pure micrococcus culture. Okay, so often that's what has to occur in medical microbiology labs. You're going to discover, you guys, when we start doing metabolic tests on Thursday, you have to have pure cultures of microbes. You can't have mixed cultures of microbes to do your metabolic tests. Also, later when we start doing antibiotic sensitivity testing, we have to have pure culture of microbes to perform those antibiotic tests. You can't do those tests on mixed cultures. You just end up with garbage. So this is another really important technique for you guys to master, is how to move from a mixed culture to pure cultures. This is a very important technique for you to learn. Okay, so with your plates, first of all, you want to ask the question, did I get isolated colonies? And remember, it doesn't matter where, you guys. If you got isolated colonies on quadrant 2, that's fine. So, did you get isolated colonies? And then secondly, you want to see if you can find evidence that you may have 2 different microbes growing in there. Now, on some of the plates, like yesterday from your colleagues, some of the plates, the serratia had totally overwhelmed, the whole plate. So on some people's plates, we couldn't find evidence of the micrococcus. But then about half your colleagues in the Monday lab, we saw a second colony growing. Okay, so just take a look at your plate, see what you think. Describe the colonies, it's just good practice. And then for cleanup, you guys, if you're finished with your plates, just make sure they're taped together with masking tape so the lids don't fall off. And you're going to put them in the autoclave bags, the biohazard containers at the end of the benches. With your tubes, you want to make sure you take all the tape off. Please take all the tape off. Just barely loosen the caps so they can vent. And then you want to put them in the proper bin in the kill area. Okay, and then do make sure you spray down your benches and wash your hands, because we have amplified opportunistic pathogens, okay? And yesterday people laughed at me when I said this. And for those of you that want to get ready for Thursday, remember we're going to start our metabolic test, Chapter 16, the lab manual. And this middle chart over there, that we're going to use that on Thursday to describe our phenol red fermentation tests. Okay, you guys, so I'll let you guys get to work. Okay, and just make sure you guys